Hello, this is Professor Young. We are beginning our journey of the urinary system, Chapter 23. In this session, we will be focusing primarily on the anatomy of the urinary system and really what the purpose or the function of the urinary system is. In the forthcoming WIMBA sessions on the urinary system, we will be looking more at the physiological response. In other words, how exactly does the urinary system achieve the purpose and the functions that it has been given to task. And so that is where we are heading with this. We are winding down within the semester, and I just ask that you continue to hold on and keep doing what it is that you are doing, and we will all see the finish end here very shortly. The structures, and when we talk about the structures, we're going to be referring to the gross anatomy. Is fairly straightforward and simple. It's probably the simplest out of all the systems that we have looked at to date. We have a pair of structures found on the dorsal side of the abdomen, and they are the kidneys. You can see that the adrenal glands are firmly attached and sit directly on top of them. And the kidneys are going to be doing the bulk of the work within the urinary system. Coming off of the kidneys and basically emptying the kidneys of their product are the ureters, which are these hoses or tubes that you see coming down, and they both empty down into this additional structure, circular structure down here at the base, which is the what I want it to do, which is the urinary bladder. Right down here, and then to remove the waste products, we have the urethra. And so we have the two kidneys, we have the two ureters, we have the one urinary bladder, and we have the urethra. And as far as the function of the system goes, the primary function or purpose of this system is to filter the blood. And as it is filtering blood, it is removing waste products, specifically nitrogenous waste. And we'll talk more about that on the next slide as we get into this. There is some ability for the kidneys to regulate the blood volume based on how much blood it is allowing into the kidneys to be filtered at any one point in time. It can have some limited success with increasing and decreasing blood volume. Which would directly impact blood pressure. If you increase the blood volume, you're going to increase blood pressure. If you decrease the blood volume, you would hope anyway that you would decrease the blood pressure. To help aid the process of regulating blood volume and blood pressure, renin is an enzyme that is released from the kidneys, and renin helps to stimulate hormonal control of blood pressure. And so that's the job of renin, and that is released from within the kidneys. There is a hormone that is released, erythropoietin, which is a hormone that is released, and we've talked about this before. We know that erythropoietin has direct control over the stimulation of red blood cell production. And so that's what we see going there. We also see calcitrol being synthesized within the kidneys. Calcitrol, and that's a term that I will throw up here for you real quick. I think I messed up the spelling of that. I did. Calcitrol is the base form of, I'm sorry, scratch that. Calcitrol, I'm rushing and it's not working. Calcitrol is a form of calcidoil That sounds better. Uh, and calcidoil is basically a form of calcitonin. And so we see this linkage between calcidol and calcitonin. Both of these, and calcidol is basically the unfinished version of calcitrol, but calcitonin we know is released in the parathyroid and helps to regulate the reabsorption of calcium within the blood. Calcidoil and calcitrol also help to do the same thing. And then the reason why they are released from within the kidneys is because the kidneys can themselves monitor to a limited extent the level of calcium within the blood because all the blood is flowing through there. And so if it's releasing, or if it's reabsorbing too much calcium out of the blood, that means they can go ahead and release calcitrol to go to the bones and help spark osteoblast activity to reabsorb all that kind of good stuff. We mentioned that one of the functions of the urinary system, the chief function, is to eliminate waste. And I made brief mention of the nitrogenous aspects of that waste. The key, or the most abundant, if you would, type of waste that we are dealing with is the formation and creation of urea. I can't believe I just did that. Of urea. Right there. And urea is a byproduct, a finished byproduct of breaking down proteins into amino acids. We know proteins are always recycled. We know amino acids are the building blocks of those proteins. Urea basically comes from ammonia. Ammonia is the direct byproduct of breaking down those proteins and back into their simpler form of amino acids. And so what ends up happening here is in the liver will convert ammonia over into urea. Urea is less toxic. The key here is less toxic. It's still toxic, but it's less toxic than the ammonia. Other forms of toxic nitrogenous waste is uric acid and creatinine. We know we at least remember, should hopefully remember creatinine when we discussed, and creatine phosphate when we discussed muscle contractions in AMP1. And creatine phosphate basically helps aid in ATP production, short-term creation of energy. And creatinine is the byproduct. It's a waste product of creatine phosphate, as it helps to make the ATP. And again, it's a nitrogenous waste. Typically, the levels of nitrogenous waste within the blood are extremely low. You have 15 1,000ths of a gram of urea found within a tenth of a liter of fluid. Extremely low amounts of urea that is found within the blood. If those levels begin to rise, that typically indicates some kind of issue going on within the kidneys, whether it's renal failure or maybe it's UTI, urinary tract infection, or maybe the person just has one kidney and the kidney can't keep up with the demands. Whatever it is, as those levels increase, it typically means that there are issues within the kidneys. When we look at the overall anatomy of the kidneys, we see the following key structures and areas. Underneath of, and basically this area right here, I'll kind of start here just to kind of get it out of the way. This area right here is the renal pelvis, and the structures that are emptying into the renal pelvis are the major calyx. And then the minor calyx are found right at the base of these upside down pyramids. And so we've got the again, the renal pelvis. And the renal pelvis can be correlated to number 1, and then we have the major calyx, which can be linked to number 2, and then the minor calyx, which can be linked to number 3 on this diagram. If we were to remove that and look at the area underneath of that area, that would be the hilum. It's kind of where everything just kind of gathers and collects and is concentrated within the kidney. The area surrounding or towards the outside is what is referred to as the renal cortex, and that cortex does tend to drip down in between these upside down pyramids that you are looking at, and that is what is referred to as the renal columns. And then we have the renal medulla, and the renal medulla is characterized by these pyramids that we see formed right here. In fact, we call them renal pyramids. It's probably one of the easiest terms that you can try to remember. Anatomists did try to make something easy for you. And so this area all in here, these are all renal pyramids. At the base of the renal pyramid, kind of where the renal pyramid and the minor calyx joins is the renal papilla. And so that kind of serves as the emptying area from the renal pyramids into the minor calyx. And then again from there, from the renal pyramids, we go into the minor calyx, the major calyx, the renal pelvis, and then the renal pelvis leads down into this little guy right here, which is the ureter. And that will then lead to the urinary bladder. And so that's the overall structure of the kidneys. And again, these are all structures that you can, for the most part, see and make out when you do a kidney dissection. What you typically cannot see, unless you have a very well injected specimen, are the vessels of the kidney. And I'm going to start from the largest, and of course the largest is going to be right here, which is the renal artery. And from the renal artery, we head into smaller vessels, which are called the segmental arteries, which are going to be highlighted right here in green. And then from the segmental arteries, we head into the interlobal arteries, which are going to come up in between the lobes, right into this area. And from the interlobal arteries, we head into the occurrent arteries, which is going to bring us right up into and around the pyramids. And then the interlobular arteries are what I will do in white, I guess. And they are the little hair-like finger projections that you see sticking up top. And so they're the various regions of blood flow that we have going through the kidney and surrounding those renal pyramids. As far as the venous system, reverse it. We have the interlobular veins going into the arteric vein, into the interlobal vein, and then there is no segmental artery. There is no segmental vein. It goes right into the renal vein, and we basically end, and here's the renal vein right there, and that's the blood flowing in. So the blood enters, goes around the renal pyramids, which you'll understand why more in a couple of seconds. The blood is then filtered out into that renal pyramid. The filtered blood then enters back into the interlobular vein, to the arcuate vein, into the interlobal vein, and exits the kidney in the renal vein. And then it will go ahead and return back to wherever it's going to be going to from there. It is inside of these pyramids where we see the nephron. It's inside of there where we see the nephrons. And that's kind of where I want to go next here, because there's two major areas to a nephron, and we have two types of nephrons. First thing I will point out, and which I think is kind of fascinating, is each kidney has approximately 1.2 million nephrons found within it. And we have two types. We have the juxtamedullary nephron and the cortical nephron. In your textbook, in almost every textbook and every diagram, this is the guy that you tend to see, the juxtamedullary nephron. And the juxtamedullary nephron is characterized by, if we were to divide this guy in half, and we're going to divide it in half right along this green line, the spot superior to it, this here is cortex, and the spot below it, this here is medulla. Well, what we see here is the glomerulus, and the glomerulus is right here. It's that circular structure right there, is found within the cortex. All of the plumbing, all of the loops, if you would, or at least the vast majority of those loops, are found down in the cortex. Whereas the cortical nephron, almost everything is up in the cortex, and only that loop of Henle is found down in the medulla. Well, within your textbooks and most of your diagrams, you see this juxtamedullary nephron. You see that structure. When the truth of the matter is, that's the least common that we have as humans. If we were birds, or if we were other types of mammals, then yeah, we would have the majority of our nephrons would be juxtamedullary. But as humans, we have cortical nephrons as the key nephron, and then then we have very few juxtamedullary nephrons. And basically, what the difference is, juxtamedullary nephrons extend down deep into the medulla because of pressure. It has a greater pressure in there, and so it can go ahead and filter more product, whereas the cortical nephrons have less of a pressure, forcing down and pressing on the plumbing, if you would, which we will get into shortly. It basically has an easier time filtering being higher up in the cortical nephron. And so that's the overall structure that we're looking at when we talk about the nephrons and what the difference is. And the difference has to be with placement. Juxtamedullary, the majority of the plumbing is found in the medulla. Cortical nephrons, the majority of the plumbing is found up in the cortex. And we will leave off with this thought today, and that is the glomerulus. And that's basically where the blood is entering into everything. It is a highly concentrated area of capillaries, and basically you are looking at the glomerulus all in this area. You can see the red blood cells as they flow through here. We have two major structures, or at least two major arteries. We have the afferent artery, which is bringing blood in, and then we have the efferent artery, which is allowing the blood to leave the glomerulus. And it is here in this area, the afferent arterial and the efferent arterial, where we see regulation of how much blood is going to be filtered. The afferent arterial, if you constrict that, you're going to reduce the amount of blood that's going to be coming into the glomerulus, and so that's going to back the blood up through the rest of the system, and it's going to increase your blood volume. Likewise, if you constrict the efferent arterial, only thing you're going to be really backing up is the blood within the glomerulus itself. But if you dilate the efferent arterial, then you're going to allow blood to flow through the glomerulus faster. And so that very, very well could decrease blood volume, especially if the afferent arterial hasn't changed much. Surrounding the glomerulus is this clear space, and you can see that clear space right there, and you can follow it right on around right there. That is called Bowman's capsule, and where you have a parietal layer and a visceral layer. The visceral layer is deep, and the parietal layer is external. and made up of epithelial cells. And basically, because you are filtering out toxins from within the blood, within this glomerulus and Bowman's capsule, you don't want things leaking. And so that visceral layer is protected by podocytes, specialized cells that have foot-like projections that are going to be there to seal up any kind of leaky junctions that the visceral layer may have. And then you have the capsular space, which is the entire outer covering. You have the vascular pole, which is going to be found vascular pole down on this end, and that's going to be because of, again, that's where the vessels are. And then the urinary pole down this end over here. And basically, the sugar, salt, and water is going to be allowed to pass through into the Bowman's capsule, as well as any nitrogenous wastes. And the emphasis here is to try to keep blood or protein and sugar in the blood vessels. And that's where we will pick up, and the next time we see each other within lecture. Again, as always, if you have any questions, please feel free to email me. I'm always available to you, or jot them down and ask me in class. This is Professor Young saying have a good night, or good day, or good early morning, depending on when you are listening to this.